Hey everyone, welcome back to the second lecture in Chapter 8. We're now going to talk about aerobic respiration. So aerobic respiration is a metabolic pathway, and it includes 4 different stages: glycolysis, transition, Krebs or citric acid cycle, and the electron transport chain. So this is a catabolic activity in which we take glucose and we oxidize it completely and we produce ATP. So the glycolytic pathway is the first pathway. It is an anaerobic process, which means no oxygen is required for this process. All living things use the glycolytic pathway, but many living things use other pathways as well. So we use glycolysis as the first step of aerobic respiration, but some organisms only use the glycolytic pathway. And so in this pathway, we're going to produce a small amount of ATP, a total net gain of 2 ATP. We're going to produce a couple of those coenzymes that we are using to oxidize the glucose. And so we form a couple NADH. And the end product that we're going to produce is pyruvate. The ATP that we do produce is through a process known as substrate level phosphorylation. So substrate level phosphorylation is a very scary looking process when you read the word, but all it is taking a phosphate and adding it to an ADP, an adenosine diphosphate. And when you do that, you produce an adenosine triphosphate. So this is not an oxygen dependent process. It's anaerobic, and for this process to occur, we have to use a little bit of ATP first. So the first step of the glycolytic pathway is an investment phase where we add ATP, and that activates the glucose. It forms a more activated glucose that then splits into 2 molecules of of glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate then are going to be converted into pyruvate as they are oxidized. So this process is going to generate a total of 4 ATP through that substrate level phosphorylation, but since we used 2 ATP in the beginning, we're actually going to only gain 2 ATP in the process. So once glycolysis is done, we have these 2 molecules of pyruvate. Pyruvate can't enter into the citric acid or Krebs cycle. So we can only have a 2-carbon molecule enter. And so pyruvate has to lose a carbon in a process known as decarboxylation. And in that process, we're also going to remove more hydrogen. So we're going to oxidize it. So the transition step is a very brief step in which we're going to take a carbon and a hydrogen from the pyruvate. And so we form 2 molecules of acetyl coenzyme A. We form 2 NADH, and we form 2 carbon dioxide molecules. And so here we have that pyruvic acid. Pyruvic acid is decarboxylated. Carbon is going to attach to oxygen, so we have to have oxygen now available for transition to occur. Here we have the 2-carbon group that's left. It is going to be oxidized, and we're going to add in coenzyme A. So coenzyme A mixes with this 2-carbon molecule, and oxidation occurs, and we form an acetyl group carried on coenzyme A. So the next stage is then going to be either Krebs or Krebs cycle or the citric acid cycle. They're the exact same cycle. Krebs is named for the individual who found it, and citric acid is named for what is produced in the cycle, but it's the exact same cycle. So in the Krebs cycle, each of the acetyl groups is going to be dropped off, and that is going to bind with another molecule called oxaloacetate. And then the acetyl group that bound to the oxaloacetate is going to be oxidized completely and decarboxylated back to the oxaloacetate molecule. In this process, we're going to have one ATP form through substrate level phosphorylation. We're going to have 3 NADH form and 1 FADH2. And remember, NAD and FAD are the two coenzymes that work in the citric acid cycle. And of course, we have carbon dioxide here, so we have to have oxygen again. So let's look at this process. Here we have that acetyl-CoA coming in. Acetyl-CoA moves in, and it binds to this oxaloacetate, forming a 6-carbon molecule called citrate, which is where the citric acid cycle gets its name. Citrate is then converted to isocitrate. It is oxidized and decarboxylated to alpha-ketoglutarate. It is then oxidized and decarboxylated to succinyl coenzyme A. It is dephosphorylated to succinate, oxidized to fumarate, converted to malate, and oxidized back to oxaloacetate. You do not have to know every one of these steps. What you do want to know is that we started with an acetyl group carried on coenzyme A. Acetyl was dropped off, bound to this oxaloacetate, formed the citrate, and then through a series of oxidation events and decarboxylation events, we produce a total for, so there's 2 acetyl groups entering, so for 2 turns of this cycle, we produce a total of 6 NADH, 2 FADH, and each of these acetyl groups has 2 carbons, so we produce a total of 4 CO2. And then because we are producing a small amount of ATP through substrate level phosphorylation, we produce 2 ATP total. And so at this point, we have only gained a net total of 4 ATP. And the cell respiration, for those of you who have already heard about this, you know that there's a lot more ATP that has to be produced. So the majority of this ATP is going to be produced in the electron transport chain. This is through a process called oxidative phosphorylation. So we're going to oxidize these coenzymes, and we're going to phosphorylate ADP. So you have to have oxygen, again, available in this process. The electron transport chain is embedded in the cytoplasmic membrane of prokaryotic organisms. It's embedded in the mitochondrial membrane of the eukaryotes, the inner mitochondrial membrane for eukaryotic organisms. And so let's see what that looks like. This is the electron transport chain. So you have these different ion pumps or channels where hydrogen can flow across. Hydrogen is moving from a lower concentration to a higher concentration, so that's against a gradient, and so it's the shuffling of electrons from one protein carrier to another that drives the pumping of the hydrogen into this outside the cytoplasmic membrane, so in this inner membrane space, okay? And then we have this ATP synthase molecule over here where we're going to produce the majority of our ATP. So aerobic respiration requires oxygen. I mentioned that. Oxygen is the final electron acceptor. We need, for aerobic respiration, we need the Krebs cycle, which also requires oxygen. We need a cytochrome oxidase, and we need oxygen detoxification enzymes. So oxygen is actually a very toxic atom. And if it is not in the form of oxygen gas, O2, it becomes what we call a free radical. And this is very dangerous to our cells. It can damage our cells. It can damage the plasma membrane. It'll bind to the plasma membrane. It can damage our DNA. It can damage the nucleus. It can cause cell death. It can cause cancer. So it's very dangerous, okay? So we have to have these enzymes. And so there's lots of organisms that can't use the aerobic respiration pathway because they don't have the special oxidation enzymes that protect them, or they don't, and so they aren't protected from oxygen gas. And so they use alternative pathways. In these alternative pathways, they use another molecule, but not oxygen, as their final electron acceptor. In these pathways, they typically don't make as much ATP either. The amount of ATP produced is going to depend on each organism, but it is exceedingly lower than aerobic respiration. So I'll let you read this on your own. This is on the PowerPoint. I'm going to move over and show you what's going on in the next slide, okay? So here you can see NADH coming in, and it's dropping off its hydrogens at this first protein. FADH comes in and drops off its hydrogens at the second protein. So NADH is oxidized, and the hydrogen ions flow through this first protein channel. The electrons move from 1 to 2 to 3 to 4 to 5 different protein groups, okay? And then they attach to oxygen, which is sitting here. The FADH drop their electrons and hydrogens here, so the hydrogens move through the second channel. The electrons only go through 4 of those carriers. Because of this, they're going to produce less ATP. And so these hydrogen ions are now in a high concentration over here. The only way they can move back down to get back into equilibrium is to flow through this ATP synthase molecule. This ATP synthase then produces ATP as hydrogens flow through, the complex spins, and ADP is phosphorylated into ATP. For every NADH that drops off its hydrogens at this first carrier, 3 ATP are produced. And for every FAD that drops off its hydrogens at the second carrier, 2 ATP are produced. And so here you can look and see the amount of ATP that is produced in each of the processes. Here you can look and see the number of reduced coenzymes that are produced in glycolysis, transition, in the Krebs cycle. And so we have a total of 10 NADH, 2 FADH that are produced. We have a total of 4 ATP that were produced through substrate level phosphorylation. And then all of these molecules, these 10 NADH, are oxidized in this pathway here. And for every hydrogen that flows through, we get 3 ATP. So we produce a total of 10 times 3, which is 30 ATP. For every FADH that dropped its hydrogens off at the second carrier, we produce a total of 2 ATP. So 2 times 2 is 4 ATP. So we get a total of 34 ATP in the electron transport chain. Does that make sense? And so we have 34 and 4 gives us a theoretical maximum of about 38 ATP. 38 ATP is the maximum amount of ATP we can produce from the oxidation, complete oxidation of a glucose molecule. So there's always a difference. The theoretical maximum is typically said to be 36 to 38, and that is dependent on if we're talking about a eukaryote or a prokaryote. In a eukaryote, we have to push the NADH and the pyruvate into the mitochondria after the glycolytic pathway. That takes energy. So we're going to have to account for that energy that we're using, and that's why we only get about 36 ATP, whereas in prokaryotic organisms we don't have to do that, so they get the full maximum. I'm going to stop here, and we'll talk about fermentation in our next video. Bye.